How's it going, everyone? Today I'm going to be presenting this paper called Soil Microbiomes Show Consistent and Predictable Responses to Extreme Events by Knight et al. So the so the bottom line up front is there's a global increase in the frequency and magnitude of extreme climate events. The soil microbiome plays an important role in soil health and regulates the biochemical processes in the soil. And the researchers sought to understand how the soil microbiome of grasslands from all over Europe would react to extreme climate events. They're kind of curious, would the soil microbiome react at all? If yes, how would it react? And would it react in repeatable and predictable ways? Um, of the extreme events tested, the heat treatment they applied produced the strongest changes, and it led to enhanced dormancy and sporulation genes, as well as a decrease in metabolic versatility. The soil microbiomes from different climates react in similar and predictable ways. This is crucial for predicting environmental consequences of extreme climate events and its effects on the soil and its microbiome. It's critical for improved land and soil management as we move forward with climate change. So, some background about climate change. Climate change is the long-lasting change in the average weather patterns. So, it's often just thought as the warming, because you often hear global warming, but it's actually all weather patterns that are included in this. And this can lead to severe consequences such as severe drought, reduced water availability, fires, flooding, rising sea levels, loss of polar ice, increase in catastrophic storms. And all of these can lead to the reduction in biodiversity across the globe. Um, Climate change is pretty heavily influenced by human factors, leading to the increase in greenhouse gas emissions, such as carbon dioxide or CO2, methane CH4, and nitrous oxide N2O. And this can have lasting effects on agriculture, food availability, animal populations, world geography, global economies, global health, and all that. And I included this chart just based on the US. It's heat wave characteristics in the US. So it's average number of heat waves per year and average uh, or the heat wave season length per days. You can see that as decades have gone on, um, we've had more heat waves per year. And as time has gone on, the length of the heat waves have gotten longer and longer. I don't know if any of you guys have experienced this, but a couple years ago in Texas, we had over 100 days, 100 consecutive days over 100 degrees. So if that is an indicator, I don't know what is. So some more background information about the soil microbiome. We're all in the master's program, so I kind of hope that we know what it is at this point. But it is the community microorganism, or the community of microorganisms that lives in the soil. And that includes bacteria, archaea, fungi, protozoa, viruses. And it's essential for the nutrient cycling in the soil, oxygen production, soil structure, health, soil health, plant health, and just the overall health of the ecosystem. And I've read somewhere, it might have been one of our lectures, about how the um, about half of our oxygen comes from microbes. So, if the soil microbiome isn't healthy, then, we're going to be impacted by the lower levels of oxygen. But a healthy soil microbiome, what is it needed for? It's needed for um, to regulate in nitrogen fixation levels. It's needed to balance soil nutrients and break down organic matter, the production of healthy and abundant crops, which is super important, suppression of harmful microbes via competition, and balance greenhouse gas emissions. Another thing that's pretty important is that it's a source of important biotech innovation, discovery of new antibiotics and enzymes. In my undergrad, I did a lot of research with bacteriophage, and those samples that we acquired and we found those phage in were actually soil and sewage samples. So there's a lot of development with like phage therapy. So soil microbiome could be a very important thing for biotech information here in the near future. So now getting into the article, um, with global climate averages deviating from the norm, and we're seeing an increase in severe storms in their frequency and their magnitude. These researchers sought to understand how the soil microbiome of the European grasslands would react to these extreme climate events. They noticed that the current knowledge on this subject was pretty lacking. Um, it was pretty narrow focused just on like single soils and single soil systems, and they hoped that by broadening the scope to like an area, or like a area of the world, rather than just like a single focus point, that they'd be able to observe patterns and how these climate events affected the microbiome of the soil, and then be able to use that for land management, soil preservation, that kind of thing. So the methodology is pretty dense. Um, I think this page is probably the most important. Um, so here are the points that they sampled from. So they sampled in 10 countries. Each country is represented by one of these circles. And then in each country, they sampled 3 sites. So that's 30 replicant sites. And then for each replicant site, they created 7 plots that are 1 by 1 meter, at least 5 meters apart, and then they collected 4 soil cores from each one of those 7 plots. Those cores were then pooled and sieved and homogenized per replicant site. So per one of these 30, one of these like these triplicate sites at each one of these 10 countries. Each replicant site was then divided into 20 pots, known as microcosms, and the total number of them is 600. So that's a lot of samples that they're having to process. In the timeline, this is S0, so down here. Um, the samples taken throughout the study were measured for soil moisture, nutrient contents, DNA based, and had DNA analysis and metagenomic sequencing done. And then those samples, so those microcosms, were acclimatized in a growth chamber for 7 days. So in this acclimatization period. These microcosms were randomly divided into 5 treatments. So control, drought, flood, freeze, and heat. And then those treatments or disturbances were applied for 2 weeks, as you can see here. And then they did testing at the end of these 2 weeks. So this is S1. And then the samples were allowed to have a 1 month recovery period. So time point S2 is 1 day into the recovery period. S3 is 8 days or 1 week into the recovery period. And then S4 is 1 month or 26 days. And to be specific about the treatments and what all that entails, so the control is 18 degrees Celsius and 60 degrees WH or 60% WHC, and WHC is water holding capacity, which is self-explanatory. It's just how much how much water the soil can hold before it's like oversaturated. The drought conditions are 18 degrees Celsius, so same as the control for temperature, but then a decreased water holding capacity. Flood, same temperature as control, but 100% water holding capacity. Freeze is -20, but the same WHC as the control. And then heat is 35 degrees Celsius at the same water holding capacity as the control. And I was curious when I was doing this at what these um, growth chambers looked like because it sounded so scientific and fancy. I looked them up and they're kind of boring. This is the exact model they used. It's literally just a big incubator. It's temperature controlled, and then they have looks like some sort of apparatus to water them without having to open and close these constantly and ruin the um, atmospheric conditions in here. So these researchers actually did quite a bit. So there's quite a bit to their methodology. Um, so this is pretty detailed. If you want to pause and read all of these like sub points, you can. But for the most part, these main ones are the most important. So they also um, measured greenhouse gas emissions, so CO2, N2O, CH4. And they also measured and analyzed dissolved carbon and nitrogen, as well as the enzymatic activity through enzymatic activity assays. DNA was also extracted from the initial soil samples, so like S0. And then the microcosm samples were also um, tested and DNA extracted. The prokaryotic and fungal community composition of the initial and microcosm samples were also analyzed at all time points, S0 through S4, and they used the V4 through V5 regions of 16S rRNA. And then they used Illumina MeSync platform as well as the Data2 pipeline in our software. The functional genes, well, the functional gene composition at the initial S1 and S4. So S1, from remembering correctly, is the end of the disturbance period, end of the treatment period, and S4 is the end of the recovery period. And they determine this through whole genome or whole metagenome sequencing using NovaSeq and Diamond Blastex. And then all the statistical analysis and plot generation was completed using our software. And I'd imagine they used RStudio, even though I couldn't really find a place where they stated if they used it or not. But RStudio is pretty common. So here is the first part of the data. And by the title, just to give you a little bit of background as we're going into this. So the sample origin is the driving factor in microbial composition. So as you can see, there's country differentiated by color. And then these are a bunch of NMDS plots. So these are non-metric multidimensional scaling plots, and they're used to visualize the relationships between complex and multivariable data sets. They also used, so these NMDS, and can never get used to saying those, NMDS plots, the distance for these are based on a Bragg-Curtis dissimilarity, which a Bragg-Curtis is used to quantify the dissimilarity between species composition or in species composition between two different sites. And these plots are pretty flexible and they're pretty common and good for use in for like ecological data and community composition like they're using here. You can see this stress number on all of them. That's a representation of the goodness of fit. So the closer to zero, the better. Zero point 0.1, so as you can see, 0.1, 0.114, 0.108. So zero point 0.1 is a pretty fair fit. It's means that your data is pretty reliable. Excellent fit is 0.05, like less than 0.05. So these are pretty solid. Um, but this is, so there's three of these. There's prokaryotic taxonomic data that was compared um, in these plots against country location, so where the samples were taken. And there's one for fungal taxonomic data and then shotgun metagenomic data. And you can pretty easily tell, so all these locations, you can tell that the composition of these microbial communities are pretty distinct based on where they're from. So you can see, like, for example, Germany. You can see in prokaryotic and fungal, it's a pretty distinct group, as well as shotgun metagenomic. You can see that they're pretty tightly together, more tightly in prokaryotic. And then Greece, same thing. They're all grouped together, all grouped together, all grouped together. Um, and these graphs are showing, like where they're located is representation of the like composition of the group. So the closer they are together, the more related they are to each other. So that distance is the pretty key factor in this. So a point here is less related to a point here than it is to one close to it. And as you can see, across all of these, location is a very big driving factor in the composition. Each one has a pretty unique composition based on location. So for this one, microbial communities show predictable shifts in response to treatments. So these are pretty similar to the NMDS plots, but they're a little bit different. So these are RDA plots. They are partial redundancy analysis plots, which is similar to a principal component analysis, a PCA. Um, I don't know if you've used those in previous labs or at work, but these generalize information from large data sets and find trends within the data. But an RDA is a little bit different than a PCA in that an RDA is constrained to an explanatory variable. And in this case, that's the applied treatment. And then a PCA is unrestrained or unconstrained. So these plots, so yet again, you have prokaryotic, fungal, and shotgun metagenome taxonomic data. These plots visualize the relationship between treatments, the color, and community composition, so where they are on the plot. They're inputting a data set of taxonomic and functional gene abundance data and then constraining it to the applied treatments, so control, drought, flood, freeze, and heat. The percentage on these axes represent the amount of variation in the data that is explained by that axis. So each axis, it's not really an axis, but like the data set, the dimension of this is explaining 5.85% of the variation and so on down the line. Um, the so like the NMDS plots, the closeness of these points is like the main thing that we're looking at here. So closer they are, more similar. The further away they are, the less similar. Um, and as you can see, heat treatment shows the greatest effect in all three of these plots, with flooding being a close second. It is most prominent in the prokaryotic plot. As you can see, they're really tightly grouped together along this line. Whereas, they're still grouped together, less so compact. And yet again, you can still see the trend, but it's much more broad. And then the flooding is the close second. So you can see here, you see the arm extending out, pretty defined, but still not as defined as the heat. And same here, and same here. So it's important to note, um, I thought it was a pretty important thing to note is um, that all of the treatments are reacting in a pretty similar manner across all of the plots, showing that the individual treatments are producing a pretty consistent and predictable response to each unique treatment that's being applied. So like heat supplying is affecting the composition in this manner away from like the relationship relative to the control, the gray points, the freeze, the blue, and then the yellow, the drought, and also the flooding over here. They're all pretty relatively similar. All of the points, all of the different treatment groups are going to the same points of the plot, indicating similar relationships to each other based on these treatments. You can see that the heat and flooding can produce fairly unique response, and that the drought and freeze produces a similar response to each other. So like flooding kind of does its own thing, heat kind of does its own thing, but then drought and freeze, you can see that they're pretty similar, even in prokaryotic and fungal and shotgun metagenomic. They're still kind of in the same place. They're pretty on top of the control, which indicates that they still had some change away from control, but it just wasn't that significant. And the researchers actually made a pretty interesting point about why these two were pretty close to being control and why they reacted in a very similar way. Well, more so why they reacted in a similar way to each other, not how they react to the control, but they kind of theorized that this was because in drought, you have less access to usable water. It's just not there. And then in freezing, you have water, but it's frozen and it's not usable. And they kind of came up with an idea that maybe it was because, lack of water that they both reacted in very similar ways. But this just goes to kind of reinforce what this title of the slide says is that the microbial communities were showing pretty predictable responses based on treatment. Heat caused them to react one way, flood caused most of the communities to react one way or like another, and then freezing and drought caused them to react and shift in a unique way as well. So this chart or these charts, I was not a very big fan of. I find them pretty confusing. And I had to spend a fair amount of time trying to understand what I'm even looking at. But this is depicting but these microbial communities started displaying 11 general strategies in response to these treatments. And if you look, oh, first, these charts are actually comparing the abundance of these things called ASVs or amplicon sequence variants. They're unique error-corrected DNA sequences derived from amplicon sequencing data. So they're similar to an operational taxonomic unit, an OTU. You might have used these in research before, might have seen them in class. Um, but ASVs have a pretty high fidelity, and I believe they kind of became in use in like 2013, I believe. I think that's kind of when they became, I think they were used in a paper and then it kind of became the standard. But they became the standard because they have really high fidelity down to like the nucleotide, and it allows for really precise comparison between samples. Because they're so precise, you can tell pretty much exactly what's there and how much of it's there. But these models show the response of um, these ASVs to the climate events and treatments. And then using the ASVs, you can determine, from what my understanding is, you could determine taxonomy from these as well. And then based off the abundance, you can kind of tell how much is there. So the center of the figures is a taxonomic tree of the 500 most abundant ASVs. This looks like a phylogenetic tree kind of thing in the center, but then it's surrounded by four rings. It's hard to see the rings, but it's easiest to see. Like here's one distinct ring, and then there's another, and there's another, and then there's an interior ring as well. Each ring represents a different treatment. The outer ring, so starting on the outside and going inward, there's heat, and then inward one is freeze, then flood, and then drought. The authors also went in and put some general categories here. So firmicutes, acidobacteria, actinobacteria, proteobacteria, those types of things. And then so on this one, it's prokaryotic ASVs, and then on the right, uh, fungal ASVs. So Asomycota, Basidiomycota, so on. Um, so one of the things that the researchers did is they measured resistance and resilience. So resistance is the ability to tolerate a treatment. That's kind of how they define it. So you subject this community to heat, and um, how well it tolerates that is resistance. And they did this by comparing the ASV abundance throughout the treatment. So they did all this testing at all the various time points, S0 through S4, and then they used those abundance values to determine, how well um, these communities were able to resist these treatments based on like the abundance. So if it started out at some arbitrary number, 0 or 1, let's say, and then at each time point it remained 1, then you could say that it is resistant. There's no change in this number. But they also measured resilience, which is the ability to recover from a disturbance. So it does change. It is changed by this treatment, but then it returns back, or at least close to kind of what it originally started out. So it starts out at this arbitrary value 1 at S0, but then as it goes on, say it increases to S3. But then by the time it's S4 again, at the very end of the recovery period, it's back down to 1, the same value that it was at S0. So it's able to change, but then it recovers back to its normal. Um, I found this, like I said earlier, I found this figure pretty hard to understand. As you can see, there's lots of different strategies down here. There's resistant, which means nothing changed, resistant with positive resilience. So overall, there wasn't much of a change, but if it did, it increased ASV abundance, but very, like ever so slightly, but then it came back down to that initial value. Um, there's resistant with a positive divergent. So it was affected and then it kept going. But then there's a positive impact of resilient, which we kind of described it in this previous one. It did change throughout the time points, but then by the end it returned back to the close or add to it. It returned back to its original value, or at least close to it. And then positive impact stable. So, the ASV abundance increased when the treatment was applied, and then it hit a level and it just kind of remained constant throughout all the time points. and then positive impact diverges. It increased off the initial treatment or at the beginning, and then it just kept increasing. And then the same thing can be, it's like vice versa for these bottom ones that show like negative resilience. All these were positive that showed positive changes in ASV, and then all these on the bottom are negative. They show like a decrease in ASV abundance. Um, and I kind of alluded to this, but orange indicates a positive change in abundance, and then this blue purple represents a negative change. And then this white is supposed to represent a resistant stable, so nothing happens throughout. But if you look at the charts, it looks gray, not white, which is pretty frustrating. Um, but yeah, you can tell like there's all these various shades. They're very subtle between this one and this one and this one and this one. You can tell, obviously, you can tell from like this darkest all the way over to this lightest. It's pretty easy to see. But when you're looking at these tiny little rectangles, and it's even smaller on the paper itself, like you really can't tell. And it makes this chart really, really difficult. And if I was to change one thing about this, I'd figure out a different way to represent this data. Um, but besides the point, whether or not I like it, the most common thing or the most common group or strategy that you can see on here is the resistant group with no change, which is indicated by this white or this gray color. And as you can see, even prokaryotic and fungal, most of these aren't changing at all. Most of them are resistant. Um, then the second most common group is the positive or negative impact that then stabilized. So it's actually most of these darker colors that you're seeing are actually following. So here on this chart and this other one, most of the darker colors that you're seeing on these rings are representing these two groups, the positive impact stable and negative impact stable, where they change, but then kind of hit a constant point. Another thing that you can notice is that you can see like certain reactions, certain strategies are kind of conserved in different groups. So you can see a lot of purple here, a lot of orange here, more purple here, and then same thing here, like orange, orange, orange. So all these like taxonomic groups, they kind of have these strategies conserved within them. Thought that was pretty interesting. So in this next slide, we are looking at the abundance of protein functions and how they correspond to climate events. So figure A, the one on those left, um, shows the effects of each disturbance, so each treatment, with the 28 highest level gene functions compared to the control down to and the control is this center line down the middle of all these treatments. So drought, flood, freeze, heat, and the control down the middle, or not necessarily a control, but like a baseline. Here are all the functions. These bold ones on the left indicate ones that had significant changes. And then same with these bold like sections here. They represent um, significant changes within these treatments. And it's pretty easy to tell. Oh, and one thing to note is that um, since there isn't a key for this, so red indicates that there was an increase in gene abundance following treatment, and blue indicates a decrease in gene abundance. That seems kind of counterintuitive since, like, in my head, red equals bad, red equals like decrease, but that's actually the opposite. So red indicates increase. And you can see that drought, freeze, flood in general had decreases in abundance. So they're all shifted to the left of the line. And then flood was different, and most of them increased or to the right of the line. But then more notably, when you look at heat, heat's kind of the ball hog and it holds all of pretty much all of the significant changes. Um, you know, all of these dark areas. So you can see that heat um, is one of the factors, like one of like the main factors for, or one of the strongest factors for causing these shifts in like gene function. And you can see that in that first plot. You can see like the on that NMDS plot, I believe that's what they're called, where heat was so strongly like pushing out from the group. You can see that here. So, but lots of these functions um, amino acid derivatives, carbohydrates, cell wall and capsule, DNA metabolism. Here's the one that was possibly affected. So you see a bump in dormancy and sporulation genes right here, and you also see it in freeze and flood. Um, then fatty acid, lipids, membrane transport, nucleoside, nucleotides, decrease in photosynthesis, protein metabolism, RNA metabolism. So you can see that the heat is really negatively affecting the ability for these cells, these microbes to function. But then you pair that with this huge bump, it's a very significant bump in dormancy and sporulation genes, which seems logical. If you're not able to function, then you at least need to be able to hunker down and survive. Figure B, so it's the number of proteins on the x-axis, and then up here it's the proportion of proteins that are affected by the applied treatment, and they did correspond to these treatments or these functions on the left. Yellow indicates that the treatment was affected. Blue shows that it was not infected. Um, Overall, there 46% of the genes in proteins were affected by the treatment. So you can see predominantly or approximately half, just roughly looking down, each one of these is roughly split in half when you look at the general trend. So, 46% of these functions were affected by these treatments. You can also see that for carbohydrates, there were a ton of proteins. Um, there are a ton of proteins related to carbohydrate utilization. It looks like 1,500, with about 45% of them affected by the treatment. So this is showing that the initial properties of the sampling sites had a pretty big impact on the resistance and resilience of these microbial communities. Um, these are Spearman's rank correlations, and yet again, it's used to um, show correlations between um, pretty big data sets. One thing to note is this correlation coefficient. So a correlation coefficient of 1 means that, I have these little examples down here, for every increase, so a correlation coefficient of 1, for every increase in this value, the other value on the other axis is increasing as well. So increase also increases, increase also increase. And then for a negative correlation coefficient, um, so you have an increase in resistance as you go along the x, but you have a decrease in this value, and it continues down. A positive correlation coefficient that is not 1 means that generally this trend is true, but you will have some where it falters. So it's just like a general plot of data that has a positive slope, and the same for like a negative like 0.5 correlation coefficient. So a general plot of data that you're used to seeing that has a trend of a negative slope, whereas the negative one is like a very defined line. Um, but the key takeaways from this figure is that both resistance and resilience, so indicated by the colors and the shades, how dark they are, is basically the same. Um, well, the big takeaway is that the resistance and resilience to heat, drought, and to a lesser degree freeze are lower in cold and wet climates. You can see that in these. So I put a key over here if you want to look at this. So for this example, like um, annual precipitation max, you can see that it's a -1, or it's close to a -1. It's like a negative like zero point 0.8 or something like that. But generally, as the value of the annual precipitation max um, increases, the resistance and resilience decreases. And then the same for like temperature max. You can see that with a really high temperature max value, the resistance also increases. And you can see this trend that they're talking about with climates can be observed here and here, and you can see that with the treatment, it's very strong in the heat treatment, both in resistance and resilience. Um, Figure B, this is enzyme activity, and then microbial substrate use, gas fluxes, and then soil carbon and nitrogen concentrations. This figure is showing the relationship between the soil function measurements on the Y and the relative abundance of functional genes for the metagenome. And then it's comparing it to a treatment and then time points S1 and S4. But it's showing that there's in fact a link between these metagenomes and the soil functions. And if the metagenomes were similar, they typically had the same function. And pretty much across the board, except for maybe S1 and floods, you can see that the enzyme activity and the ability to use various carbon sources is pretty strongly correlated with the metagenome of the soil. So to wrap things up, the soil microbiome is a key player when it comes to the overall health of soil, and it helps regulate the various biochemical processes that occur there. We know that there's an observed global increase in the frequency and magnitude of extreme weather. Climate change is a problem. It's here, and it seems to be here to stay. To better understand the effects that these global climate changes or global changes in the climate have on the soil microbiome, researchers collected a very wide range of data regarding the soil, the initial sampling sites, and the soil microbiome itself. And it was found that the location that the sample came from was a very large driver in the composition of microbial communities, with each sample taken from the same country exhibiting very similar composition to each other. It was also observed that the microbial communities shifted in similar ways in both composition and function when applied to the same climate events. So a total of 11 different strategies were found and observed in response to these events, showing a varying degrees of resistance and resilience to these events. And then the properties of the initial sampling locations also played a pretty big role in the resistance and resilience of these microbial communities, with resistance and resilience being higher in areas with warm and drier climates. So the soil microbiomes around the world, regardless of what climate they are subjected to, will react in similar predictable ways to these extreme climate events. Extreme heat caused the strongest changes in composition and function of microbial communities. And even though it was determined that soil microbiomes will react in similar ways, having the details about the initial sampling site, the local microbiome composition, soil properties will allow for greater prediction. And lastly, some questions. So, does this study's reliance on metagenomic sequencing and annotation lead to accurate conclusions, or are the researchers exaggerating their findings? If there is any part of the experimental design that you change, what would it be? And then how would it improve the experiment? And then even though these observed effects were small, do you think they are ecologically meaningful? Will they have a positive or negative impact over time? And then the study tested samples in the lab rather than in the field. What are the trade-offs of testing in the lab under artificial conditions rather than in natural conditions in the field? And then how might environmental organizations utilize the results of this study to better manage the environment? And here are my references. And thanks for watching and listening.